Dear children, welcome to online classes of biology. So today we'll start a new chapter, that is chapter 20, locomotion and movement. So I hope you understood the previous chapter and have studied well. Okay, so in this chapter only few topics are there for you to study, that is about the skeletal muscles. Okay, then about the structure of skeletal muscles or muscle fiber. Okay, then mechanism of contraction of muscles. So this much only you need to study. Okay, so you have studied that there are three types of muscles, isn't it? We have also seen in the lab, okay, different types of muscles like skeletal, isn't it, or smooth. Okay, skeletal, smooth and cardiac muscles. These are the three different types of muscles, or you can say striated, unstriated and cardiac muscles, isn't it? Skeletal muscles are what? They are striated. They are also known as striated. Why they are known as striated? Because they are having stripped appearance, means dark and light bands can be seen in them, isn't it? And as they are controlled, okay, under our that they can work under our control, they are also known as what? Voluntary muscles, isn't it? So as the activities of them are controlled under the voluntary control of us, our nervous system, they are also known as what? Voluntary muscles. Okay, and they are attached to the skeleton, the skeletal system, so they are also known as skeletal muscles. So skeletal muscles, stripped muscles, voluntary muscles, all these are same only. Okay, so when you see the structure, we have studied the structure of skeletal muscles, where we have studied in the tissues chapter, isn't it? Animal tissues. So there I have told you, you can see they are what? Not having tapering ends, they are dark and light bands, you can see. So all this speciality we have studied. And when you see the smooth muscles, okay, they are also known as visceral muscles, because they are located in the inner walls of the hollow visceral organs, isn't it? Like what? Alimentary canal, reproductive canal, etc. And they are not under the control of our nervous system. They are also, so they are also known as what? Non-voluntary muscles, isn't it? And they are not having any striations, so they are also known as smooth muscles, isn't it? So likewise, skeletal muscles and visceral muscles I have told you. Now what are cardiac muscles? Cardiac muscles are found only in heart, isn't it? And they are having what? Branching pattern. Okay, and they are what? They are stripped only, and they are involuntary in nature. Okay, so all this we have already studied. So we have to study especially about the skeletal muscles and their what? Structure and function in this chapter. Okay, so skeletal muscles, okay, they are forming a bundle. Okay, a number of muscles, skeletal muscles, they form a bundle. Okay, muscle bundles or fascicles, and they are commonly they are bounded by, they are what? Covered by a collagenous connective tissue called fascia. So in our body, the skeletal muscles are seen as what? Bundles. Okay, as a number of bundles, which are, okay, muscle bundles, or what we call them as fascicles. Okay, you can see here in the diagram. So these fascicles are held together by a common collagenous connective tissue known as fascia. Okay, and each muscle bundle or fascicle, if you see, you can see a number of muscle fibers. Okay, muscle fiber is actually what? It's a cell only. Muscular cell is known as what? Muscle fiber. As they are elongated cells, we call it as what? Muscle fiber. Understood? And each muscle fiber, okay, it's a cell only, animal cell only. So it will have what? A plasma membrane, a cytoplasm, etc. Isn't it? So the membrane, plasma membrane of muscle fiber is known as what? Sarcolemma. So what is sarcolemma? It's the plasma membrane of the muscle fiber. What is muscle fiber? It is a cell of a muscle tissue. Okay, skeletal muscular tissue. Okay, consists of what? muscle fascicles, which are what are this muscle fascicle? Group of muscle fibers which are surrounded by a collagenous connective tissue called fascia, and in each fascia or muscle fascicle, what is there? Number of cells called muscle fibers. So muscle fiber is an animal cell, okay, which is having a plasma membrane, and that plasma membrane is known as sarcolemma, and the cytoplasm is known as what? Sarcoplasm. And you can see many nuclei are situated in them, so they are also forming a syncytium. So what is syncytium? Is a cell with a number of nuclei, okay. And you can see in the cytoplasm there is some endoplasmic reticulum. It's an animal cell, isn't it? So endoplasmic reticulum is there, and we call them a special name known as sarcoplasmic reticulum, because in the functioning of this muscles, okay, they play an important role, and they store what? Calcium ions, okay. And the muscle fiber, okay, they contains, okay, number of parallelly arranged filaments, okay, proteinaceous filaments, and these filaments are known as myofibrils. So you can see here in this diagram, you can see each of this is what? A fascicle, okay, and in that many muscle fiber is there, and each muscle fiber, if you see, it will have a sarcolemma, then sarcoplasm, that is the the membrane, and sarcoplasm is the cytoplasm, and in that you can see what? Sarcoplasmic reticulum, and you can see a number of parallelly arranged protein molecules, and they are known as what? Myofibrils. Is it clear? Or myofilaments. And each myofibril or myofilaments is having what? Alternate dark and light bands, okay. So why this dark and light bands are there? That is the portion where they are having actins, they are light in nature, okay. And wherever they are having myosin, there they are having what? Dark what bands, okay. So parallelly you can see, okay, along the length of the muscle fiber, you can see in the sarcoplasm there are certain rods, okay, rod like structures, proteins arranged. They are known as what? Myofibrils or myofilaments. And in the myofilaments or myofibrils, you can see dark and light bands. These dark bands are actually corresponding to a protein known as myosin, okay, and the light bands are actins. Understood? Now the I band, okay, that is light band is also known as I band or isotropic band, and dark band is also known as anisotropic band, okay. Means A band means thick band contains a protein called myosin, and thin band contains what? Actins, okay. And they are arranged as rod like structures parallel to each other, and also to the longitudinal axis of the myofibrils, okay. Actin filaments, I told, they are thin, okay, and they are known as what? I band, okay. In the center of each I band there is a line known as Z line. Understood? So you can see here in this diagram it's more clear. That is you can see A band, okay. A band is actually this, understood? And you can see I band, this is I band, understood? And here also in this diagram also it's very clear, that is this is dark band and this is light band, okay. And in the light band there is a in the center of the light band there is a region known as Z line, okay, Z line, okay, which bisect the I band. And the thin filament are firmly attached to the Z line, and the thick band is known as what? A band. So this is the I band, this is the I band, okay, and this is the A band. And in the center of the I band, what you can see? Z line. And in the center of A band, what you can see? M line. Understood? So A line is there, M line is there, Z line is there. Is it clear? The A and I bands, they are arranged alternatively throughout the length of the myofibril. So this is what a myofibril. Okay, and you can see they are arranged alternatively. Understood? And then, and the portion of the myofibril between two successive Z line is known as the functional part of the skeletal or the skeletal tissue, and it is known as the sarcomere. So what is sarcomere? Between two adjacent Z line. Okay, the part between two adjacent Z line is known as what? Sarcomere. So this much part is actually what? The sarcomere. This is one Z line, this is another Z line, and the part between these two is known as what? As the functional unit of contraction, and is known as what? Sarcomere. So it includes what? Half of one A band, a full full A band, okay, a part of I band, okay, half of I band, then a full A band, and the part of another or half of another I band, isn't it? So this much is known as what? The functional unit of contraction, and it is known as the sarcomere. So what is sarcomere? The functional unit of contraction, and what it includes? It includes half of, okay, I band and a full A band and half of another I band. This much forms the functional unit of contraction and is known as what? The sarcomere. Okay, so you can see that, understood, in resting stage, the edges of thin filaments on either side of the thick filament, okay, you can see here, okay, this is what? This is the A band, okay, this much is A band and this is I band, and this is the Z line. Okay, so you can see that in resting stage, the edges of the thin filament on either side of the thick filaments, okay, they overlap the free ends of the thick filaments, okay, and they will not move over or arch over this much part. This much part is known as the edge zone. Okay, so when the muscle is in the resting position, understood, these are arranged one after the other, isn't it? One A band will be there, then one I band, then again A band, likewise. But you can see in the resting stage, the edges of the I band, okay, they just overlap, okay, over the ends of the A band, but it will not but it will be leaving a space. So this space, okay, which is left by the I band is known as the edge zone. Okay, edge zone, and you can see it clearly here. That is, sarcomere includes what? The distance between two adjacent Z line. So Z line is what? The center of the I band. Is it clear? So like this they are arranged. Okay, now let's see the structure of contractile proteins. So each actin filament, okay, is made up of two filamentous actins, okay, which are helically bound together. Okay, you can see each of this is what? A F actin. Okay, this is one F actin and this is another F actin. Okay, so each what? Actin filament is actually made up of what? Two F actins which are helically bound each other. Okay, and in each F actin you can see what? G actins. Okay, you can see it's a chain made up of what? Globular proteins or G actins. Is it clear? Now, two filaments of another protein known as tropomyosin. Okay, tropomyosin. Can you see this? Okay, so this is the tropomyosin. Understood? Tropomyosin also run along the this length of the actin filament. Okay, then a complex protein known as troponin, you can see here at regular intervals. Okay, so these all are what? Troponins. Okay, troponins are also distributed, okay, at regular intervals on the tropomyosin. Understood? And in the resting stage, means when the muscle fiber is not in action, okay, that is the actin filaments, okay, will have what a subunit of troponin that mask means what, which covers the active binding site for the myosin on the actin filament. So something you understood, that is what we are learning is, I told you, the muscle fibers, okay, or myofilaments. Muscle fiber is what, the cell, isn't it? And it is having sarcolemma, sarcoplasm, sarcoplasmic reticulum. And then I told you, in the sarcoplasm there are what, rod shaped proteins, okay, they are known as rod shaped structures. They are known as what, myofibrils or myofilaments. And myofibrils and filaments are made up of what, two types of proteins. What are they? Actins and myosin. Understood? And they are arranged alternate, okay, to each other. Means once here, if actin is there, then myosin, then actin, likewise, okay. And I told you, in the resting stage, actin filaments will arch over the what, the myosin filaments, or A band, isn't it? A bands, okay. So A band contains what, myosins, and I band contains what, actins. Understood? So all that is very clear. Now what we are studying is this actins and myosins structure, ultrastructure we are studying, okay. So in that I told you, each actin filaments, what they are made up of, they are made up of two what, F actins, okay, filamentous actins. You can see here, isn't it? It's very clear. 1, 2, okay. Likewise, two what, actins are there, which are helically wound, okay. And then what is there? Tropomyosin, that is also two are there, okay. So two are there which are helically wound, okay. That goes along with the with the F actins. And in the tropomyosin, okay, you can see some proteins are there. They are known as troponins, okay. They are found at regular intervals. You can see here, okay, and here. And what they do is, they cover the active site for what, for the myosin filaments. Myosin filaments will come and bind. I will tell you when it is, okay. When muscle contraction happens, this has to be, okay, removed. This troponin has to be removed. Then what will happen? The active site will become free, and into that only myosin will come and bind. Understood? So like this it is arranged. Now myosin filaments, if you see, each myosin is also a polymerized protein made up of monomeric proteins. Polymerized means what? Which are made up of monomer units. And its monomer is known as what? Meromyosin. What is it? Meromyosin. Understood? Now, meromyosin, it is having two important parts. Okay, a globular head with a short arm is there. Okay, that is known as the HMM. Understood? And then they have a tail. Okay, a tail will be there, and then what? This two part. Okay, head region and a arm region. This two together we call it as what? That is HMM. Okay, heavy meromyosin. And the tail part is known as what? Light meromyosin or LMM. Okay, and the HMM component, that includes what? The head and the arm. They just project outward at regular distance and angle from each other from the surface of a polymerized myosin filament. Okay, so this much part is forming what? The crow's arm. Okay, and the globular head is having an ATP, means it is actually an active ATPase enzyme itself, and it is having what? A site for ATP, ATP to get attached. Okay, so this crow's arm, okay, this is part of what? Myosin. Okay, so this myosin will go and bind to where? To this region. Okay, that we are going to study. So this much structure of actin and myosin, if it is asked, okay, you have to explain this. Actin's structure includes what? They are made up of two F actins which are helically wound to each other. Then two filaments of another protein called tropomyosin also runs close to the F actins. Okay, and a complex protein called troponin is distributed at regular intervals. Okay, so you have to explain about F-actins, and F-actins are actually made up of what you can see. Each F-actin is made up of globular, round shaped structures. They are known as G-actins or globular proteins. Okay, or globular actins. Understood? So then you have to say about what? F-actins. Then you have to say about tropomyosin and troponins. This many are there in what? Actin. And myosin consists of what? They are polymers of what? Myromyosin. Each myromyosin is having what? A head region, an arm, short arm that forms what? HMM, and a tail known as LMM. Okay, HMM component, they form what? Cross arm with the actins. Okay, and this head region is having what? ATP binding site for actins. Okay, now let's study about the mechanism of muscle contraction. So mechanism of muscle contraction can be explained by the sliding filament theory. Okay, so according to this, contraction of muscles, okay, happens by the sliding of the filaments over the, means actin filaments, thin filaments. Okay, they slide over the what? Thick filaments. Okay, so do you remember this? I told you when the muscle is in the resting stage, you can see that actin filaments ends are just arching over the what? Myosin filaments, isn't it? Myosin filaments, you can see here, this is the A band and this is the I band, consisting of what? Actin filaments. So actin filaments, what ends will be arching over, it will be just projecting over the A band. Understood? Now according to the sliding theory, okay, sliding theory states that the sliding of what? Thin filaments over the thick filaments brings the contraction of muscle fiber. Okay, and for this some some signals are needed, and this is sent by the central nervous system, and when this messages reaches to the muscle fiber, okay, there are some junction between the motor neuron and the sarcolemma, and that is known as what? Neuromuscular junction or motor end plate. Means the brain will decide, okay, means you have to contract that particular muscle, then signal will be sent from the central nervous system, isn't it, to that muscle. So where it will be received in the that is in the neuromuscular junction. Okay, so at that time what happened? A neural signal reaching this junction, okay, they releases some neurotransmitter. Okay, neurotransmit means some chemicals will be released, like what? Acetylcholine. Okay, and this causes an action potential in the sarcolemma, and this will be reaching to the Sarcoplasmic reticulum. I told you already, sarcoplasmic reticulum is having a major function in the muscle contraction. Okay, because they stores calcium ions. Okay, so what happens? This signals when it reaches, that is the chemical, when it reaches to the sarcoplasmic reticulum. Okay, then what happen? It causes the release of calcium ions into the sarcoplasm. So when calcium level increases in the sarcoplasm, what happened? The subunit of troponin on actin filaments. Okay, means on the subunit of troponin on actin filaments are having some I told you, some masking effect. That masking effect will be removed, means troponin will be just removed from there. Understood? So what will happen? Using ATP's energy, okay, myosin head is what? Part in the myosin or thick filament. Myosin head will go and bind to the active site, because active site was already masked by what? Troponins, isn't it? We learned that here, isn't it? Troponin is having some masking some active site, which is removed by the calcium ions. Calcium ions, what number increases, concentration increases in the sarcoplasm, means what will happen? The troponins covering effect will be removed. Then what will happen? This will help the myosin to bind to the exposed active site of the actin, and they forms what? Cross bridge. Like this you can see cross bridge will be formed. Understood? And this that pulls the attached actin filaments towards the center of A band. Do you remember A band is having what? A band is having some zone called H zone. Okay, so towards the H zone from both the sides it will be pulled. So what will happen? The A band, okay, A band will remain same, but the I bands will get contracted, isn't it? And the Z line attached to this actins are also pulled towards inward, and that causes the shortening of sarcomere. Do you remember what is sarcomere? That is between two adjacent Z line, what is there? Sarcomere is there, isn't it? So that causes the contraction. Understand? So the during shortening of the muscle, okay, or contraction, I band get reduced but A band retain the length. Okay, so which band is retaining the length? A band, but I band getting reduced. Okay, and myosin, what happens to the myosin? They release ADP and inorganic phosphate, and they go back to the relaxed state. Now a new ATP will be binding to the cross bridge. Okay, ATP binds and the cross bridge will be broken. Okay, and the ATP is again hydrolyzed by the myosin head, and the cycle of cross bridge formation is continuing like this. Okay, and that causes what? Further sliding, that is actin filaments and means cross arm formation, and it's breaking. Okay, that will continue, and that causes what? Pulling of this actin filaments or thin band towards the inside. Okay, this process will continue till when? Till all the calcium ions are pumped back to the sarcoplasmic reticulum. Understood? Again what will happen? So once calcium ions is not available, troponin will go and bind to the active site and covers it. Okay, so that causes the return of Z line back to the original position, and that is relaxation happens. Okay, so the repeated activation of the muscles can lead to what? Accumulation of lactic acid, okay, due to what? Anaerobic breakdown of glycogen, and this is causing what? Muscle cramp. We have already studied this in 10th class. Understood? So muscles contains a particular oxygen storing pigment that is known as myoglobin, and the muscles which are having this myoglobin will be having a reddish appearance, and that kind of muscles are known as red fibers. Okay, and they contain what? Enough means plenty of mitochondria, and they are what? Aerobic muscles. They use what? Oxygen oxygen. Okay, they can store oxygen in the myoglobin. Understood? So like this what? They are aerobic type of muscles. Okay, and there are some other muscles where this myoglobin's content is less, and they will be what? Appearing pale or whitish, isn't it? So they are known as white fibers. Okay, and they have less mitochondria, and they are they do their work by anaerobically. Okay, they depend on anaerobic process for energy. So this is the topic that you have to study this chapter. So what we have learned is about the skeletal muscles. I told you what are skeletal muscles. Okay, and what is the speciality? They are also known as what? Striated muscles. They are known as voluntary muscles, and each muscles, okay, muscles are made up of a group of muscle fibers, muscle bundles or fascicles, which are what? Or group of muscle fibers. They are bound by what? Collagenous connective tissue called fascia. Okay, and each muscle bundle contains what? Muscle fiber, and muscle fiber is a cell only, animal cell, elongated cell having plasma membrane, cytoplasm, then what? Sarcoplasmic reticulum, and they all are having different names. And in each sarcoplasm, you can see rod shaped, okay, structures. They are known as what? Myofibrils, and each myofibril is having what? Dark and light bands. Okay, and the dark band contains what? Myosin, and the light band contains a particular protein known as actin. And we have learned, okay, this actins and myosin, they are arranged alternate to each other. That only we have seen in all these diagrams. Understood? And then I told you I band is having a center place known as what? Z line, and this A band is having a central line known as M line. Okay, and in between two Z line, that forms the unit of contraction, and that is known as sarcomere. Okay, and we also studied about what? The different, okay, we studied about the different actions, like mechanism of muscle contraction, okay, structure of contractile proteins. All that is different questions. You have to study that separately. Okay, each of this muscle contraction, how it happens, and this is explained by which theory? Sliding theory. Okay, the sliding filament theory explains what? The muscle contraction. Okay, and how it is happening I told. Okay, when signal comes, neural signal comes to the neuromuscular junction, what happens? It produces a neurotransmitter, and that will generate a action potential in the sarcolemma, and that causes the calcium ions to release into the sarcoplasm. Understood? So increase in calcium level leads to what? Binding of calcium ions to what? Troponin. So troponin will be just removed from there. So what will happen? That sites will get exposed. Then what happen? The myosin will go and bind to there, and a cross arm will be, or cross bridge will be formed. Isn't it? That pulls the what? Actin filaments towards the center of the A band. Understood? So A bands will remain what? Same, but I band is getting shortened. Okay, so all this is very clear, I think, and from this chapter only you need to study this much what I have taught. Thank you, children.